The goals of this assay are to dissect the role of disease-relevant processes in blood-brain barrier coverage disruption, identify the underlying mechanisms, and test potential treatments in vitro.
This method can answer key basic and applied research questions in the blood-brain barrier and neurodegeneration fields. For example, by addressing whether disease-relevant stressors disrupt preformed network structures and identifying the underlying pathways. Through further screening, treatments can be identified that prevent stress-induced blood-brain barrier disruption for future in vivo testing.
The implications of this technique extend towards mechanistic understanding of and treatment for Alzheimer's disease because of the importance of the homeostatic functions of the blood-brain barrier on cognition.
Though this method can provide insight into blood-brain barrier dysfunction in Alzheimer's disease, it can also be applied to other neurodegenerative disorders with blood-brain barrier dysfunction, such as stroke.
Begin by passaging HCMECD3 immortal endothelial cells at a ratio of 1:5 by incubating the cell sheet with calcium and magnesium-free HBSS for 3 minutes. Then detach the cells using 5 milliliters of 0.25% trypsin EDTA for 5 minutes. Neutralize the trypsin by adding 5 milliliters of EBM2 complete medium. Collect the cells and centrifuge at 290 times G for 5 minutes at 4 degrees Celsius. Following centrifugation, discard the supernatant and resuspend the pellet in EBM2 complete medium.